Hello and welcome to my fourth video on cell biology, and this time I'll be talking about membrane transport. And in order to do that, I'll be talking about the different types of transports. So, for example, active and passive transport. Then I'll be talking about diffusion and facilitated diffusion, osmosis, osmolarity, and osmotic potential. And then lastly, cytosis, so exo and endocytosis. I hope you enjoy. First of all, cell membranes have been mentioned a lot throughout this topic because they're an integral part of membranes and a very interesting one at that. And to refresh our memories, cell membranes are semi-permeable, so they only allow certain materials to cross freely, mostly those that are small and lipophile or hydrophile. And then cell membranes are also selective, so membrane proteins may regulate the passage. Not always, but sometimes. Moving on, here we have a graphic of the different kinds of transports within the cell membrane. And starting out with the active transport, we see that there are two kinds of active transports, namely one, the primary and the secondary. And primary ones are pump mediated, so they're so-called ion pumps or just in general membrane pumps. The most common ones are, for example, sodium pumps or potassium pumps, and they through ATP carry or open through voltage gated or ligand gated mechanisms and let the specific ions into the cell or out of the cell. And the pump mediated ones, as we can see here, need ATP. And we have, for example, uniport, that's one single gradient, so from low to high. So uniport meaning that only one material is transported. And then in the primary pump-mediated ones, we also have co-transport. And as you can see here, there are two different concentrations, so that means there needs to be a different kind of material that exists in a higher concentration on one side and a lower concentration on the other side, and respectively so for the other one. Then we also have secondary active transport, that's carrier-mediated. And I'll be talking about carrier proteins a bit more when I talk about facilitated diffusion, but in general there are integral glycoproteins that do something called a conformational change, and through a passive through an active process without ATP though, let in or out specific materials, as you can see again here. Then we also have passive transport that does not require materials and is along the concentration gradient, whereas active transport is against the concentration gradient and makes direct use of metabolic energy. But here we have the three different types of passive transport I'll be talking about in the following. So facilitated diffusion for uniport and simple diffusion. And here, when we talk about diffusion, we define it as the net movement of particles from regions of high concentration to regions of low concentrations. It's a passive process and it takes place until an equilibrium is reached. That means when on both sides of the semi-permeable membrane the same amount or the same concentration of that material consists there. And across cell membranes, simple diffusion takes place mostly for small nonpolar molecules, as I mentioned earlier. And there are different factors that affect the rate of diffusion. For example, the steepness of the gradient, so how big the difference in concentration is, the distance moved, so for example how big the membrane is, or how big the area in general is, which is also another factor, so surface area is involved. Another factor could be the barriers to diffusion, so what kind of proteins the materials, for example, in facilitated diffusion would need to use in order to pass the membrane or cross the membrane. Then we have also temperature as factors of the diffusion rate, because temperature affects the kinetic energy. And then lastly, we also have molecular size as a factor of diffusion rate because of the dynamic nature of molecules and how fast they could move. Right. As I mentioned before a couple of times, there are two different types of diffusion. So when we have the simple diffusion, that is a movement directly through the membrane without any assistance, that takes place, for example, in cell respiration. We also have facilitated diffusion, which is the passive movement across cell membranes via membrane proteins for molecules that cannot move across freely. So if molecules that can move across freely are small nonpolar molecules, that means that molecules that cannot move across freely are big and polar. And for those, there are two different types of proteins they could use. And if you would like to update your knowledge on membrane proteins, you can check out the last video I made on the structure of the membrane, where I briefly talked about membrane proteins. But without further ado, we start with channel proteins, and and this process is also called a channel-mediated facilitated diffusion, where an integral lipoprotein contains hydrophilic pores via which inorganic ions pass through the membrane. These channel proteins are also ion selective, and they may regulate passage according to certain stimuli. It also propagates the movement along the concentration gradient, and it's the faster one of the two, so it's faster. It has a faster rate of transportation than carrier proteins. And talking about carrier proteins, here they are. Carrier proteins and their process is called a carrier-mediated facilitated diffusion, and it involves integral glycoproteins which bind the solute and allow large lipid insoluble molecules to pass that cannot normally cross the membrane or cannot cross it freely. They undergo the aforementioned conformational change, which you can see in the picture on the left-hand side. And this conformational change takes place to translocate the solute across the membrane. It only binds specific molecules, which is similar to the process of the substrate-specific enzymes, like a key lock principle, and they may move molecules against the concentration gradient as opposed to channel proteins, but that occurs only with the assistance of ATP, so energy, metabolic energy. It has, because of the conformational change and because it's more work, it's a slower transportation rate than channel proteins. Moving on, we have osmosis, which is a special kind of diffusion or a special kind of passive transport because it is a passive process. So osmosis is defined as the passive net movement or diffusion of water molecules from a low solute to a high solute concentration across a semi-permeable membrane. And we also have the characteristics of water, which is an universal solvent. That means that it associates with polar or charged molecules, forming a so-called solute. Water also equalizes to solutions when they cannot cross a cell membrane, so that is where the equilibrium takes place. And if there's a higher solute concentration, that means there are less free water molecules. And the same thing you can see here in this graph from BioNinja. So the low solute concentration, we can see a lot of free molecules, free water molecules. And on the right hand side, we see a high solute concentration where they associate with polar or charged molecules, and they form these solute molecules, therefore, and you can see that the number of free molecules decreases dramatically. And because of that, because there's now a low solute concentration and a high solute concentration, the concentration of the net movement has taken place. So the osmosis has already taken place in this one. And when we talk about osmosis, there's also a mention of the so-called osmotic potential or osmotic pressure. And it's defined as the tendency of water to move into solution of the dissolved solute. And if there's a high solute concentration, that means there's also a high osmotic potential. And it's also called water potential. So that's what you would understand under osmotic polarity. Then you also have the so-called osmolarity, which is the measure of solute concentration number of osmoles of a solute per liter of solution, and that's written as a unit of osmol or osmoles per liter, and that can be investigated in different environments and in different experiments. And the three states that a cell could be in are here as follows, the three states of osmolarity. And starting out with the hypotonic osmolarity, it occurs when there's a high solute concentration on the outside and a low solute concentration on the inside, which means that the cell, because of the net movement along the gradient, that means that the cell will absorb H2O in pure, if it's in isotonic pure water. And in animal cells, this has the effect that they are lysed. That means they swell and they could eventually burst because they have a less stable structure because of the lack of a cell wall. And if you want to remind yourself of the ultrastructure of cells, you can watch the first video on the ultrastructure that I have posted. We have plant cells which become turgid in a hypotonic state. That means that the cytoplasm inside expands. Then we have the kind of neutral state of cells, namely the isotonic. That means that the inside and the outside, the extracellular and the intracellular space have an equal solute concentration, and they will remain in equilibrium, and both the animal cell is normal and the plant cell is normal or flaccid. Then we also have a hypertonic osmolarity. That means that the solute concentration outside is low and the solute concentration inside is high, which, because of the concentration gradient, the cell will lose water in isotonic conditions, and the animal cell in this case undergoes a process of crenation, or it will shrivel, as you can see here, as the water leaves. And in the case of the plant cell, it becomes lysed or plasmolysed. As you can see here, the cytoplasm breaks down to some extent, or undergoes the same, is lysed as an animal cell would be in hypotonic conditions. And yes, those are the three different kinds of osmolarity, or the three different states of osmolarity. Then we come to the last topic, cytosis. And cytosis is an active process that most cells do, and it happens when the plasma membrane folds around a substance to transport it across the membrane, whether in or out. And that depends whether it's exocytosis or endocytosis, and it depends on the kind of material and the requirements of the cell. And it's a form of function of flexibility of the membrane, and it's therefore bulk transport through localized cell activity of the cell cytoskeleton. And here are the two different kinds of the cytosis, starting with the exocytosis, exo like export, and it means that the cell is expelling material, and this material is exported in vesicles or vacuoles, which I've also mentioned in my video on the ultrastructure of the cell. In order to be exported, the material fuses, or the vesicles fuse with the plasma membrane and diffuse out into the extracellular space, and thus they are expelled. And this happens in so-called secretory cells, or secretory cells, I don't know. So cells that secrete a certain material. And an example for that could also, for example, be the process of synapses. So exocytosis is also there when the vesicles, the synaptic vesicles, fuse with the presynaptic membrane and they diffuse out into the synaptic cleft. We also then, on the other side, have endocytosis, which is basically the uptake of material, so the opposite of exocytosis. And this is the cell engulfing material. And endocytosis is part of the plasma happens when a part of the plasma membrane is pulled inward and surrounds the liquid or solid material. If it's a liquid material, it's called pinocytosis, and if it's a solid material, it's called phagocytosis. And yeah, it's this plasma that surrounds the material is moved from the extra to the intracellular space, and there the material is enclosed in vesicles and drawn into the cell and used accordingly. This process happens in, for example, protozoans and phagocytes. And this was the video on the topic of membrane transport. Thank you for watching and have a nice day.